So I'm going to be following up the theme that Tal already started, which is diseases of pyrophosphate metabolism. My lab has been working on bone mineralization disorders for many years, and trying to understand the molecules that control bone mineralization led us to understand what happens during ectopic calcification, soft tissue calcification. And it happens to be the same molecules we've been studying all along. So it was very natural for us to focus on vascular calcification as a major medical problem that affects soft tissues. And that's what I will be talking to you about now. And I will try to make it short and sweet because we're all waiting for coffee. So I'll be brief here. So there are two types of vascular calcification. Very important to clarify from the beginning. I am not talking about atherosclerosis. And most physicians do not distinguish intimal from medial calcification, so I want to make sure you understand the difference. It is not atherosclerosis, it is not intimal calcification, atheroma formation, calcification of the atheromatic plaque that I'm talking about. I'm talking about calcification of the medial layer of the arteries, the muscular layer, which allows arteries to comply when blood passes through. It needs to move up and down when the blood passes through. So when you have ectopic deposition of bone here, you essentially affect the ability of these arteries to comply. You essentially have bone forming in the arteries. That leads to hypertension, leads to cardiac hypertrophy, and essentially these patients die of heart attacks and cardiovascular disease. That is what I'm talking about today, okay? There are a number of monogenic defects that lead, rare diseases, that lead to medial vascular calcification. Here are at least 4 genes that have been associated with that. There is something common to all 4, which I will mention in just a brief moment. Here is a deficiency in an enzyme that produces inorganic pyrophosphate, an enzyme that is involved in transport. Maybe this is even easier to see. Production of pyrophosphate, transport of pyrophosphate, transport of adenosine. Again, pyrophosphate metabolism is involved here. Alkaline phosphatase, my favorite molecule, is involved in clearing pyrophosphate to phosphate, and phosphate transporters. All of these molecules here that are involved in the biology of vascular smooth muscle cells are the same molecules involved in the biology of the skeleton, okay? So what we've learned from the skeleton can be translated to the biology of vascular smooth muscle cells. Why am I studying rare conditions? Because they are a wonderful window, another speaker already said this, wonderful window to understanding metabolic pathways and how these molecules work. And as you saw from hypophosphatemia, focusing on a rare disease could be a very quick path towards an IND approval that then the drug could be used for a more general condition. So here I'm showing you a rare condition known as generalized arterial calcification of infancy, where kids essentially die within 6 months of age. They are treated, although not perfectly, by bisphosphonates. There is room for therapeutic improvement. And then here I show you a major medical problem in the form of end-stage chronic kidney disease, where most patients that are going to hemodialysis have medial vascular calcification, and essentially patients in dialysis do not die of their kidney condition, they die of cardiovascular disease, okay? So a serious medical condition. Here is one example of a calcified artery in the foot, which was published in this review by Dwight Towler, an expert on vascular calcification. And look at the title of this review of his. It's an imminent disease epidemic, under-recognized by the medical community, that has a tremendous financial impact on our healthcare system because, for example, in type 2 diabetes, the cost of amputations caused by this vascular calcification passes all the other costs associated with other manifestations or other cardiovascular diseases. So this establishes significance. This is a significant problem. Now, pyrophosphate metabolism. We need a certain amount of pyrophosphate for controlled calcification. As Hal mentioned, pyrophosphate is a potent inhibitor of calcification. If you have too much of it, you have hypophosphatemia. You're essentially suppressing calcification where it is needed, okay? If you have too little, then you're allowing calcification to take place where you don't want it, in soft tissues, including the vasculature. So arterial calcification is a problem of low pyrophosphate concentrations, all right? So what are the molecules that control pyrophosphate? Well, primarily there is an enzyme that produces pyrophosphate from ATP. There is a transporter, the ankylosing molecule, that transports pyrophosphate from inside the cell to the extracellular milieu. And then the enzyme alkaline phosphatase, tissue non-specific alkaline phosphatase, whose function is to cleave pyrophosphate and form phosphate. In so doing, it helps drive calcification by two mechanisms, contributing phosphate and also decreasing the concentration of pyrophosphate. All right? So I will show you that absence of any of these two molecules, like happens in some children with generalized arterial calcification, leads to these massive depositions of hydroxyapatite. Here you have von Cosa staining, black staining for hydroxyapatite in both mice deficient in NPT1 or deficient in the ankylosing protein. What we found some years ago was that not only do you have low systemic levels of pyrophosphate, you have upregulated levels of alkaline phosphatase by vascular smooth muscle cells in culture. These cells hypercalcify. They have increased levels of alk-phos that you can quantify, essentially a doubling of alkaline phosphatase activity. So this means that the upregulation of alkaline phosphatase is causing a further sinking of pyrophosphate concentrations right there in the vasculature, helping drive the deposition of bone in the wrong place, bone mineral in the wrong place. We found the same in uremic rats, a model of end-stage renal disease. Actually, we have an upregulation of alk-phos, as you see here, these black bands, high alk-phos activity compared to wild-type aortas. So uremic rats or these two models of insufficiency of pyrophosphate metabolism, they all have something in common, TNAP upregulation. Now, this TNAP upregulation has been reported by all those rare defects that cause generalized arterial calcification of infancy. All the authors in their papers mention upregulation of alk-phos as part of the pathophysiology. So we are convinced that all of these different rare and common diseases funnel through one common effector molecule, which is upregulation of alkaline phosphatase. And that's our target. We feel that if you can inhibit alkaline phosphatase activity in the vasculature, you can pump up the levels of pyrophosphate and suppress ectopic calcification. And that's essentially the bottom line of what I'm trying to tell you today. And it's summarized here. You have this medial calcification, normal situation. You have upregulation of alk-phos, sinking of pyrophosphate levels, and deposition of hydroxyapatite in the vasculature. So the therapeutic approach is that we propose that inhibitors of alkaline phosphatase pyrophosphatase activity can be used to prevent and perhaps even reverse vascular calcification. So in order to prove their point, we decided to see whether alkaline phosphatase upregulation was not only necessary, but could be sufficient to cause vascular calcification. So we decided to generate a mouse model, a very robust mouse model, upregulating alkaline phosphatase only in the vasculature, and see what would happen, all right? And this is what we did. We essentially introduced the human alkaline phosphatase cDNA in the HPRT locus, in the mice. It's an X-linked gene. So male mice are going to have only one X chromosome, only one gene. Females are going to have two. Now this mouse is normal as such. The gene was placed in such a way that it's flanked by LOXP sites that need to be cleaved by a Cre recombinase to activate the transgene. So when you cleave these LOXP sites, upregulation of alk-phos happens, and you have high expression. If you don't breed these mice to a Cre-expressing mouse, nothing happens, and these mice are normal. And that's exactly what we've done. So we've taken these mice that are otherwise normal, we bred them to transgenic mice expressing Cre recombinase under control of a vascular smooth muscle cell-specific promoter. And we get male mice as well as female mice, okay? Again, you can anticipate already that male mice that are hemizygotes, they have only one gene to be activated, are going to be fully penetrant in their phenotypic expression. Female mice, because of mosaic expression of X-linked genes, are going to have much milder phenotype, and this is exactly what we see. So what is the phenotype? Remarkable. You have upregulation of alkaline phosphatase and massive calcium deposition here, stained by alizarin red staining, which stains for calcium, okay? 30-day-old mouse aorta. Very young mouse. Another one, descending aorta, alkaline phosphatase staining here, hydroxyapatite deposition, look at this, all stained with calcium. Massive deposition of hydroxyapatite. Von Cosa staining for phosphate. Massive depositions, both the ascending, descending aortas. Von Gieson staining for elastin, you see very, very aberrant staining because you have all these depositions here. And what are these depositions like? Well, they essentially are bone being formed in the aortas. And these changes in gene expression show us that. We have massive upregulation of alkaline phosphatase at 14 days and 30 days of age. This is the human transgene that we have upregulated. But upregulation of this essentially causes a sinking in pyrophosphate levels that I told you, that leads to all of this. Leads to upregulation of endogenous mouse alkaline phosphatase, RANKS2, which is a transcriptional factor that determines osteogenic lineage. You have phospho-1, another enzyme involved in mineralization of bone, osteopontin, collagen-1, typical marker of bone formation, BMP2, phosphate transporters, MPP1, and all of this is upregulated. What this means is that essentially the vascular smooth muscle cells are being transformed into bone-forming cells in the arteries. Okay, that is the pathophysiology of what is happening here. Look at some x-rays. 30-day-old mouse, you see this calcified tube, should not be there. This is wild type, and here you have massively calcified aorta after 30 days in the male mouse. Alizarin red staining, this is the calcified aorta right there. And this image tells it all. This tube should not be there, should be nothing. Massively calcified aorta, even the valves, the heart valves are calcified here. Here, a cross section, calcified ascending aorta, descending aorta, massive calcification. These male mice die within 60 days of age. Okay? So essentially, the disease is so severe that they do not survive. What do they die of? Look at this slide here. Amazing. Tremendous hypertrophy after just 30 days of life. And if you slice this heart, you see that essentially you have signs of necrosis and calcification here at the tip of the heart. Essentially, they die of a heart attack. Okay? Cardiovascular failure due to this massive calcification. So, back to our hypothesis. Producing potent inhibitors of the pyrophosphatase activity of alkaline phosphatase could be used to prevent and treat this severe condition. We spent many years mapping the binding of inhibitors into the active site of alkaline phosphatase. Levamisole is a known drug, a poor inhibitor though. We mapped it. Theophylline is another approved drug, poor inhibitor. We mapped it. L-homoarginine as well. All of this work helped us understand the nature of the active site of the enzyme that could help us drive medicinal chemistry efforts towards optimizing a good inhibitor. Here another image that shows you that the pocket, you can essentially fit things in, and there are two residues in the surface that could be used to accommodate residues and side chains. Using the extraordinary facilities that we have at the Conrad Previs Center for Chemical Genomics at the Sanford-Burnham Medical Research Institute, as Michael Jackson showed you all the robotics, we have been able to do massive screening. We've screened 325,000 libraries. We got tons of inhibitors and activators, and essentially, after a lot of work, came up with some scaffolds that showed promise. They looked good. We knew that they were uncompetitive inhibitors. We were able to map them to some of the sites that our site-directed mutagenesis work had anticipated would be good sites for docking, etc. And this is a critical slide where I show you that particularly two scaffolds here are very good at preventing calcification of vascular smooth muscle cells. So you put these cells in culture, you add the inhibitor, and you prevent them from calcifying. So good effect in vitro. This is one of the inhibitors that we like very much, could do what 1 millimolar levamisole does at 10 micromolar concentration. Great improvement over a poor inhibitor. And this was a critical experiment where we could put aortic cultures, aortas ex vivo in culture, and prevent essentially calcium loads by adding the inhibitor. So here with the use of this scaffold, we can prevent aortic calcification in organ culture. So all of this looked good, and we came to a full stop 2 years ago. And we realized that one thing is to develop probes, and the other thing is to develop drugs. Okay, and this is a good probe, but it's not a good drug. So we run into solubility issues and plasma shift issues. And this is where we went back to our chemists, and they helped us turn a bad compound into an excellent drug that we can now use for preclinical and clinical work. And they use their magic to turn this scaffold, mutagenizing and changing by chemistry all these potential residues, and try to improve the solubility, plasma shift, and availability of this compound. To make a long story short, they went through this kind of array multiple times, synthesizing compounds, testing them, and so forth. And essentially, they turned this, which was a good scaffold but a bad drug, into this, which they tell me is an excellent drug, and they're ready to put this in mice as well as in humans. And if you look at the properties, everything in red are properties that are poor, and everything in green here are very good properties for a drug candidate. The IC50 has to improve tremendously. Here you have microsomal stability improved tremendously, availability improved tremendously, exposure, remarkable improvement from 1,000 to 84,000. They tell me that comparing these two compounds, we have 1,000-fold improvement in the properties of these compounds. So essentially, we have what I think is a good drug. We are now at the stage where in the next 6 months, we're going to do the preclinical studies that would hopefully take us to an IND. We have this robust mouse model that I just showed you, and we have two versions of this mouse model. We have the male mice that die young, meaning that we can evaluate not only the efficacy of the compound on growing mice, meaning developing arterial calcification mimicking the infantile disease, but we can also look at side effects on bone mineralization. That is a concern with growing children, of course. Here we have a mouse model within 2 months of age. They are continuing to grow their skeleton. We can assess the efficacy on the vasculature and side effects on bone. And then we have the female mice that are developing vascular calcification much slower. They do not have this massive hypertrophy. They do not drop dead. And these female mice are going to help us evaluate what happens when you inject this compound in adults, where your bone turnover has slowed down. You essentially have homeostasis of bone, maintenance of bone. So we have two models that I think are going to be quite exciting for us to look at. Once we have evaluated in our robust model, we look at chronic kidney disease models, ectopic bone formation, and the MPP1 model of deficiency, which is one of the rare conditions that I have already mentioned to you. So possible clinical uses of this, well, clearly, I'm very excited about the potential of using this on generalized arterial calcification of the infancy. A rare disease, there are very few children around, but you only need, as we saw, 10 children to document efficacy and to move to other aspects of the disease. Chronic kidney disease, end-stage renal disease is a huge market potentially for a drug like this. Heart valve calcification, heterotopic calcification, major problem for war veterans coming back from the wars around the world. And the Department of Defense is putting a lot of effort into trying to find treatments for heterotopic calcification. Paget's disease, Kawasaki disease. These children, it's a disease that is really not, the etiology is not known, but sometimes if you're not diagnosed properly, when a child reaches age 20 years of age, they have massive coronary artery calcification and they just simply drop dead. So another potential therapeutic use. And even osteoblastic bone metastasis. Prostate cancer is one of those tumors that metastasizes to bone, promoting bone formation, in difference to breast cancer that turns to have osteoclastic bone formation, so destroys bone. In the case of prostate cancer, you form bone, and this is what causes pain in prostate cancer patients. So this could be a treatment not of the cancer, but it could ameliorate the suffering of these patients undergo because of tremendous pain. So I think we are at the stage where I need to thank support from NIH and tell you that this is a highly collaborative project. Members of my laboratory that I need to thank, and of course all the chemists at the Institute that have made this project possible, as well as Charlie O'Neill from Emory University, who has the rat model of the end-stage renal disease. Thank you very much. Questions? Yes.
So it doesn't matter what the underlying cause of the ossification is.
Because everything seems to funnel through osteogenic conversion of vascular smooth muscle cells.
Yeah.
And part of the osteogenic conversion is upregulation of iron.
So even like this FOP treatment?
Yes, I believe it is. We need to do this, but I believe so. And that is an extreme case of heterotopic calcification. Either case, just some variations, could benefit from this. FOP, that's a very serious condition.
And do you put any of the animals?
But again, I wanted to stay away from atherosclerosis because it's a completely different beast. In atherosclerosis, you have inflammation as a major component. Here we have no inflammation. We have convergenic, osteogenic transdifferentiation of vascular smooth muscle cells.
But you have ossification occurring in the plaques?
There is no plaque to talk about. There is
no, I'm saying if you did it.
Okay, well, but see, now you're entering into an arena that is very complex. And if you talk to cardiologists, whether it would be good to decrease calcification,
it might be the wrong direction.
Precisely. They would argue, for days about whether it's good or bad. So another beast, we don't want to touch it. We have plenty to try to treat with this approach. Yes?
Can you explain how bisphosphonates like citronate help put calcium and phosphate into bone for osteoporotic people, but then it reduces?
Can you repeat the question up there?
Sure.
Thanks.
So the question is, what is the effect of etidronate, both on the vascular calcification and in the bone?
Because they seem the opposite.
Well, etidronate is an early generation bisphosphonate. It's one of those bisphosphonates that binds very avidly to hydroxyapatite. But it also, so it prevents more deposition from taking place, okay? And it also inhibits osteoclast in bone tissue, so it inhibits resorption. So from one side, you have the effect directly on osteoclast that inhibits resorption, so allows bone to essentially maintain its density, its bone mineral density. On the other hand, you are adding it because of its ability to decrease more deposition in the arteries and the ability to remove hydroxyapatite from the arteries. Now, the problem with bisphosphonates is that, first of all, it has this secondary effect on bone that you want to try to eliminate. And it is a rescue treatment. You are trying to essentially remove the hydroxyapatite that has already taken place in the arteries. The arteries have already gone through chondrogenic osteogenic differentiation. The damage to the artery is already done. You're removing the calcium and the phosphate from there, but the damage is already done. What I'm hoping this treatment will do is to prevent the damage to occur in the first place. You're stopping it at the point where they're just going to go into osteogenic differentiation. You're stopping them right there. And I think there is a window of opportunity for doing this early on in the development of arterial calcification. And perhaps if you intervene for a few weeks, you could stop this process from happening, and then you don't need the treatment for life.
Is it going to help, like in the third trimester?
I don't know. We need to begin to do preclinical trials with the model as we currently have it, and then see what the difficulties are and what the possibilities are.
I had 2 patients with this.
Right. The problem
Seems
is
to be getting better.
yes, it will get better. The problem is timing the etidronate treatment. If you go on too long, they develop secondary hypophosphatation. In fact, I'm in a paper together with Michael White, where this patient came to him because it was a hypophosphatation. It turned out to be arterial calcification treated too long with etidronate. Okay, I think we need to go for coffee. Thank you very much.